Imagine you could take a single photograph of a chaotic, just a totally superimposed crowd of people.
Right, like everyone's standing right on top of each other.
Yeah, exactly.
Right.
And then mathematically unmixing that image so you can suddenly see 4 completely distinct, perfectly clear groups of individuals. I mean, it sounds like a magic trick.
It really does. But it's not magic, it's actually math.
Which is why we are here. Welcome to the deep dive. Our mission today is to thoroughly unpack this May 2026 paper from Nature Methods. It outlines exactly how an AI tool called MicroSplit is pulling off this exact illusion, but, with the fundamental building blocks of life.
Right, it's doing it at a microscopic level.
Yeah.
It truly is a remarkable piece of computational biology.
Oh, absolutely. And for you listening, today we're going step by step. We're going to dissect the biological problem, the actual AI architecture, the empirical evidence, and the hard limitations, too. We're getting into all the details.
Because to fully grasp what this paper achieves, you really have to untangle the optical physics it's bypassing, right?
Mm-hmm.
And the specific variational splitting encoder-decoder architecture it uses.
Well, let's start with that physical roadblock then. Anyone who works with multiplex imaging knows about the triangle of frustration.
Oh yeah, the classic triangle.
Right. So picture a literal triangle in your head. You've got speed on one corner, spatial resolution on another, and light exposure or, cell health on the third.
And you are constantly trapped making a trade-off between them. You literally cannot maximize one point of this triangle without heavily sacrificing the others.
Yeah, and the absolute currency that governs that entire triangle is what we call the photon budget.
Exactly. I like to think of a photon budget like a monthly cellular data plan for your smartphone.
Oh, that's a great analogy. So you only have a finite amount to spend.
Precisely. When you're dealing with multiplexed imaging, say, you want to see the nucleus, the microtubules, and the kinetochores all in the exact same cell you're dealing with, overlapping excitation and emission spectra.
Which means the colors bleed together, essentially.
Right, we call that crosstalk or bleed through. So the traditional way around this is strictly sequential imaging. You capture one highly filtered channel, then the next, then the next.
But every time you snap a picture, you are burning through your data plan. You're draining that photon budget incredibly fast.
It's a massive drain. And once you exhaust that finite resource in a living biological sample, you just hit a wall.
Because you either have to drop the light levels, which gives you just grainy, noisy data that's practically useless, right?
Yeah, or you do the opposite. You crank up the laser to get a perfectly clean image, and you induce severe phototoxicity.
You just cook the cell.
Literally, you end up altering the very cellular biology you're trying to observe, or you just kill the subject entirely.
So we're stuck in this frustrating loop. We want richer multi-target data, but doing it sequentially either leaves us with garbage data or a dead cell.
And that physical limitation is exactly what makes this paper so fascinating.
Because they didn't try to invent a better lens, right? Or a less toxic fluorescent tag. They just bypassed the hardware limit completely with software.
That is the paradigm shift here. They approached a physical optical bottleneck purely as a computational problem.
Okay, let's do a quick little recap for you before we move on. We're going to do this in bullets to keep it structured. Bullet 1, multiplexed imaging requires sequential exposures, which totally drains your photon budget.
Right. And bullet 2, that triangle of frustration, forces you to make trade-offs between speed, resolution, and keeping the cell alive.
Exactly. And bullet 3, overlapping spectra, basically limit how many structures you can actually tag without getting bleed through.
Which brings up an interesting question to think about.
Yeah. Have you ever tried to stretch a really limited resource too far in your own work? Keep that specific feeling of frustration in mind as we introduce the AI solution.
Because that solution is MicroSplit.
Yes. Enter MicroSplit. This deep learning-based computational multiplexing method is just wild.
It really changes the game.
To visualize how it works, I actually like to use an audio analogy. So imagine you were trying to record a 4-piece band. You've got drums, bass, guitar, and vocals.
But you only have one single microphone.
Right. One mic, smack in the center of the room. You hit record, and you obviously just get one chaotic, totally superimposed audio track.
Everything is overlapping.
Right. But MicroSplit is like an AI that takes that single muddy audio file and flawlessly isolates the drums, the bass, the guitar, and the vocals into 4 completely distinct, crisp audio channels.
That translates perfectly to the imaging application, actually, except it's doing it with overlapping cellular structures.
So how does that look under the microscope?
Well, MicroSplit allows a researcher to image up to 4 completely superimposed structures in a single fluorescent channel.
Wait, a single channel, just one shot?
Just one shot. You can open up your filters, capture a broad emission band all at once, and take that visually complex combined image.
And then the AI just mathematically untangles it.
Exactly. Yeah. It computationally unmixes it into distinct individual image channels. And here's the critical part. It doesn't just separate them. It heavily denoises them in the process.
Oh, wow. Which means we essentially just hacked the photon budget. By ditching those restrictive sequential filters, we're not just saving time, we're reclaiming our entire budget.
Precisely. You're no longer discarding precious photons just to avoid spectral bleed-through. You suddenly have this massive surplus.
And you can spend that surplus however you want, right?
Yeah, exactly. You can use it to image much faster, which is revolutionary if you're tracking dynamic live cell events.
Because the cell is actually moving.
Right. Or you can image much more gently to keep the cell alive longer. Or use that budget to image even more target structures at the exact same time.
Okay, time for another quick bulleted recap. Bullet 1, MicroSplit separates up to 4 superimposed structures from just a single channel.
Bullet 2, it effectively bypasses the traditional physical limits of the photon budget entirely.
And bullet 3, broad emission filters can now be used without worrying about bleed-through artifacts ruining your data.
So here's a question to mull over. If you could suddenly do 4 tasks simultaneously using the energy of just one, what would you do with all that surplus energy?
Keep that in your back pocket. But right now, I need to push back a little bit on this microsplit thing.
Okay, let's hear it.
If we are throwing up to 4 completely different structures into one single visual bucket, isn't the AI just looking at a giant, muddy, overexposed blob?
Well, it looks like a blob to us, sure.
So how does the AI actually know what it's looking at without just hallucinating the data? I mean, AI hallucinations are a massive problem.
That is the crucial question, and it brings us right to the algorithm's architecture. How does it resolve the overlap without making things up?
Yeah, exactly. Let's get into the mechanics.
The system behind MicroSplit is called a Variational Splitting Encoder-Decoder network.
That is a mouthful.
It is, but it's essentially a hybrid. It's the child of 2 distinct, pre-existing AI frameworks that the researchers sort of spliced together.
Okay, let's unpack those 2 parents. The first one is a framework called MicroSplit, right? Spelled with the Greek letter mus.
Right, and the key thing MicroSplit brings to the table is something called lateral contextualization, or LC.
Lateral contextualization, how does that work?
Well, it's what prevents the AI from getting lost in the weeds. If you're looking at an incredibly dense overlapping image patch, you can't determine what a specific pixel belongs to just by looking at its immediate neighbors.
It's like trying to identify the architecture of an entire cathedral by just staring through a magnifying glass at a single brick.
That's exactly it. You need to see the whole building. So MicroSplit forces the network to pull back and see the cathedral.
How does it actually do that mathematically, though?
It takes the primary high-resolution input patch, but then it systematically injects image context at increasingly lower pixel resolutions deeper into the network.
Oh, I see. So it's basically feeding the AI the macrogeometry of the cell alongside the micro details.
Exactly. By understanding that broad spatial layout, the big picture, it can make highly accurate semantic decisions about overlapping pixels at the high-resolution level.
And the paper outlines 3 meta-architectures for this, right? Lean LC, regular LC, and deep LC.
Yes, so if you're running this in the lab, you can essentially choose your horsepower.
They scale up in GPU memory demand, but they also scale up in how cleanly they unmix the data.
Right, but MicroSplit is only half the equation here. The second parent framework is called denoised split.
And denoised split is arguably doing the heaviest lifting, isn't it?
It really is. It brings sophisticated, unsupervised denoising to the architecture based on advanced noise models.
Which is vital because in the real world of biological imaging, you almost never have perfectly clean, high-signal data to train an AI on.
Exactly. Denoised split allows the network to learn from and operate directly on the noisy, low-exposure data we are actually trying to process.
And that balance becomes incredibly clear when you look under the hood at the actual math that trains this AI.
Oh, the loss function.
Right, the loss function. They use a specific formula to balance these 2 tasks during training, and it works kind of like a sliding scale.
Right. Without reading literal lines of code, the formula applies a specific weight, a variable they call W, to balance the denoising task against the unmixing task.
So it's essentially the overall microsplit loss equals W times the denoised split loss plus 1 minus W times the must split loss.
And they deliberately cranked that dial to set WAM at 0.9.
Which means they are putting 90% of the mathematical emphasis on the unsupervised denoising capability.
And only 10% on the lateral contextualization splitting. That weighting is extremely telling.
Well, if you're in the lab right now staring at an overexposed cooked sample, you know exactly why they did that.
Because handling the inherent photon shot noise in biology is actually a much harder obstacle than identifying the geometry of the shapes themselves.
Exactly. If you can't strip away the noise, the semantic shapes are completely lost anyway.
And the empirical evidence of this working in tandem is just staggering.
Let's look at the case study they ran. They used the HTLIF24 dataset, imaging the nucleus, the microtubule network, and kinetochores.
Those are the tiny protein machines that pull chromosomes apart, for anyone who might need a refresher.
Right. So they fed a MicroSplit an incredibly brief, very noisy 2-millisecond exposure.
An exposure time that would traditionally give you a practically useless static-filled image.
But the network took that single 2-millisecond exposure, unmixed the 3 structures, and denoised it so aggressively that the final output was actually higher quality than a raw image taken with a 5-millisecond or even a 20-millisecond exposure.
It completely outperformed the standard baseline.
That absolutely blows my mind. We are talking about a mathematically proven 3x to 10x reduction in the required photon budget.
You're extracting vastly superior data with just a fraction of the light.
Okay, let's pause for another recap here. Bullet 1 MicroSplit uses lateral contextualization, which comes in lean, regular, and deep models to see the big picture.
Bullet 2, the loss function, heavily weights the denoise split component at 90% to ensure powerful, unsupervised denoising.
And Bullet 3 empirical data proves up to a 10-fold reduction in necessary light exposure.
So looking at that math, here's a question for you. Why do you think the developers felt the need to weight the denoising function at 90%? What does that tell us about biological data?
It's a great point to ponder, but I want to circle back to my earlier skepticism.
The hallucination risk.
Exactly.
Yeah.
If this AI is doing 90% of its work filling in gaps and stripping away noise, the hallucination risk feels massive.
It does sound risky, yes.
Like if a chatbot hallucinates a historical fact, it's annoying. But if a bioimaging AI hallucinates a structural anomaly that isn't really there, that's catastrophic for the downstream science.
The authors were acutely aware of this, which is why MicroSplit is built on a variational architecture.
Meaning it has a built-in, I don't know, mechanism.
Exactly. It explicitly quantifies its own uncertainty.
This is honestly my favorite part of the paper. How does it calculate doubt?
It utilizes something called posterior distributions. So a standard deterministic AI looks at an image and just gives you its single best guess.
Right, it just gives you one answer.
But MicroSplit doesn't do that. It learns an approximate posterior distribution of possible solutions.
So it's basically rolling the dice.
Right. When it looks at an overlapping set of pixels, it effectively rolls the dice multiple times, sampling many different possible ways to mathematically unmix that specific area.
So it's like asking a panel of 100 experts to independently draw what they think the unmixed structures look like based on a blurry photo.
That's a perfect analogy. And if all 100 experts draw the exact same microtubule filament in the exact same spot, the AI is highly confident.
But if those 100 drawings are totally different, the AI realizes it's just guessing based on the noise.
Exactly. By analyzing the variance, the disagreement between all those internal sample solutions, MicroSplit calculates a spatially resolved prediction error.
And in practice, what does that look like for the researcher?
It outputs a literal heat map overlaying your data. Where the heat map glows bright, the AI is explicitly telling you, hey, I am highly uncertain about the unmixing in these specific pixels.
Which is incredible because it allows you to confidently flag, manually review, or just completely discard unreliable data. You aren't just blindly trusting a black box.
Right. And to prove that you can trust the areas where the AI is confident, they ran a graded evidence test.
So yeah, they brought in 3 top human analysts to manually segment the images, right?
Yes, and then they compared the human-to-human disagreement against the AI's predictions.
And the validation there was pretty definitive.
It was. The natural variability between the AI's unmixed output and the target data was entirely within the normal range of inter-observer variability.
Meaning, the difference between the AI and a human expert was no larger than the difference between two top human experts just disagreeing with each other.
Precisely. It performs at a human expert level.
And because it's so surgically precise at isolating these learned structures, the researchers realized it actually had a bonus application.
The artifact eraser.
Yes. They found they could use it as an intelligent eraser for imaging artifacts, like if your sample is polluted with spurious fluorescent puncta, just glowing background trash, you can actually train MicroSplit to treat that specific artifact as a valid target structure.
Right, it unmixes the visual trash into its own isolated channel, and then you simply delete that channel,
leaving your actual biological data perfectly pristine. It literally turns a frustrating biological bug into an algorithmic feature.
It's brilliantly elegant.
Okay, recap time. Bullet 1 MicroSplit calculates spatial prediction errors based on the variance in its posterior sampling. It gives you a literal heat map of its doubt.
Bullet 2, its accuracy rivals the agreement rate between top human experts.
And Bullet 3, it doubles as a tool for unmixing and removing structural image artifacts.
So ask yourself this. How crucial is it for the AI tools you use in your own life or work to explicitly show you their level of uncertainty?
It's a huge trust factor. But let's pump the brakes again. No tool is perfect, right?
Definitely not.
And to really wield a tool like this, you have to acutely understand where it fails. The authors outline 5 specific hard limitations, and we need to walk through them.
Absolutely. The first limitation is intensity skew. If you superimpose 2 structures, and one is exponentially brighter than the other, the AI struggles deeply.
Because the sheer mathematical weight of the bright signal simply drowns out the faint signal.
Right, and the network just fails to unmix the weaker structure.
That makes intuitive sense. Number 2 is extremely low signal-to-noise ratio. We know it leans heavily on denoised split, but if your raw data is literally just static, if the signal is completely buried, the model degrades.
You can't pull a rabbit out of an empty hat.
Right. What about the third one, structural similarity?
This one is critical for experimental design. If you superimpose 2 identical-looking structures, for example, 2 different but visually identical microtubule networks, MicroSplit cannot separate them.
Why does it go blind there?
Because it relies entirely on structural priors. The AI hasn't learned to untangle overlapping string, it's learned to tell a string apart from a ball.
Ah, okay.
So if you give it 2 overlapping shapes that share the exact same geometry, the math cannot find a differentiating morphological variable to grab onto. It needs distinct spatial patterns.
Which ties perfectly into the fourth limitation, lack of spatial correlations. If the structures you're analyzing have totally random, unpredictable geometry every single time you look at them, the network struggles to build a reliable model.
Doesn't know what it's supposed to be unmixing.
Exactly. And finally, the fifth limitation involves size constraints.
Right, the network operates within a specific receptive field size. If the biological structures you're trying to unmix are massively mismatched with the input patch size, either way too huge for the AI to see the whole shape, or far too microscopic to register performance drops significantly.
And there is a massive flashing red light warning the authors attached to this tool. Do not use MicroSplit for downstream biological tasks that rely on measuring precise, absolute pixel intensities.
There's a vital distinction to make. MicroSplit is a semantic unmixer. It is not a photometric measuring device.
It's getting the shape, the location, and the morphology right.
Exactly. But because of the heavy denoising and the variational sampling, the exact absolute brightness of a specific pixel might be slightly shifted by the algorithm.
So if your experiment relies on knowing exactly how many photons hit a specific receptor, this is not the tool for you.
Definitely not.
To put all of this into context, though, we have to look at the competitive landscape. If I'm a researcher, why would I use MicroSplit over existing state-of-the-art computational multiplexing tools, like, say, the Picasso system?
Well, Picasso is fantastic, but there is a fundamental operational difference. Picasso requires multiple sequential images with known spectral overlaps to calculate its unmixing math.
It relies on a stack of input data.
Exactly. MicroSplit requires only a single superimposed image.
Which changes the game entirely for live cell imaging. If your cell is moving, taking a stack of images means your subject has shifted between frame 1 and frame 4. A single image freezes that moment perfectly.
And furthermore, the empirical comparisons in the paper show that MicroSplit vastly outperforms Picasso when the data is highly noisy.
Because of that 90% weight on the denoising.
Right. Picasso's math degrades much faster under high noise conditions than MicroSplit's architecture.
Okay, final recap of the day. Bullet 1 MicroSplit struggles with extreme intensity skew, identical structures, and measuring precise pixel intensity.
Bullet 2, it outperforms Picasso by requiring only one input image and excelling in high noise environments.
So given these limitations, how would you design an experiment to ensure your cellular structures have enough visual contrast for the AI to work?
It requires some forethought, for sure.
It does. So stepping back and synthesizing this whole journey, we started with the rigid physical limits of light, right? The photon budget and the triangle of frustration have dictated the pace of microscopy for decades.
They really have.
But by combining variational splitting encoder-decoders, basically blending lateral contextualization with massive unsupervised denoising, we now have a system that can semantically unmix biology.
We can map uncertainty with posterior sampling, match the accuracy of human experts, and drastically reduce the light needed to see the invisible.
As long as we respect the boundaries of structural similarity and absolute intensity, of course.
It represents a profound philosophical shift in imaging. We are no longer relying solely on the physics of lenses and the chemistry of fluorophores. We are leveraging the mathematics of deep learning to see clearer, faster, and gentler.
And that leads us to a provocative final thought for you to take away today. We've spent this entire deep dive talking about looking inward into the cell.
Right, the microscopic.
But if we can now semantically unmix wildly complex, superimpose, highly noisy structures from a single image channel, where else in science could this exact variational splitting architecture be applied?
Oh, it's a fascinating question.
Yeah.
I mean, could this exact math be used to untangle massive, superimposed optical data from deep space telescopes trying to separate overlapping galaxies?
Or what about medical imaging? Could it separate interwoven, noisy neural signals in functional MRI scans of the human brain?
Or even isolate specific overlapping environmental markers in single-channel satellite imagery of Earth?
I mean, the math is there, and it clearly works on extreme noise. The code is out there. The only limit now is where we point the algorithm next.
Truly.
Take those questions with you. Keep examining the hidden layers of the data in your own world, and we will catch you on the next deep dive.